everybody to our Saints talk by Professor Len Thomas. Counting Critters, St Andrews research at the interface between statistics and global wildlife conservation. Basically, how maths is saving our world. Professor Len Thomas is Professor of Statistics in our School of Mathematics and Statistics and a member of the Interdisciplinary Centre for Research into Ecological and Environmental Modelling, CREEM. Together with his team, he produces outstanding research which continues to have global impact in protecting endangered species worldwide. His game-changing software, Distance, has become the industry standard to measure population of species. This software is free, as is the training that goes with it. Although we are seeking funding for this currently, so if you're interested in supporting, please get in touch. On average, it's downloaded over 3 times a month and used in over 115 countries. And through it, there have been remarkable outcomes in the conservation and management of diverse species, ranging from whales, seals, elephants, ants, trees, and flowering plants. Professor Thomas's research has yielded lasting impacts for marine mammals worldwide in mitigating the adverse effects of naval sonar operations. Collaborating closely with the US Navy and international naval forces, he identified a critical issue: marine mammals' severe response to low-frequency sonar sound. Consequently, the US Navy changed its operating procedures, as did the Norwegian and Netherlands navies and NATO. And these are just 2 examples of a vast array of successes. In this talk, Len will explain how we can tell which wildlife species are doing it okay and which are of conservation concern, and how can we tell if conservation measures are effective in restoring depleted populations. He'll highlight the work of CREEM in developing methods for estimating wildlife population size and how new data collection technology, including remote cameras, microphones, and even environmental DNA, can potentially be harnessed to make this work even more effective. Professor Thomas will talk for about half an hour, and then he would be delighted to take your questions. We'll aim to finish at 6:15 sharp. So, Professor Len Thomas, thank you so much for joining us, and over to you.
Great. Thank you very much, Tanya. And hello, everybody. Thank you for the opportunity to talk with you and talk a little bit about what we do. So, as Tanya said, I'm going to highlight today some of the work of CREEM, which is the research centre that I work with in. And I'd like to emphasise from the outset that all the nice things Tanya said are not really me. It's part of an enormous team of people that work very closely together on these problems. We're jointly between the School of Maths and Stats, Biology and Computer Science, and I've just highlighted here our joint directors. So David Borch is from Maths and Stats, which is my home school, and Katrina Harris from Biology. Also, just while I have the picture up, this is Steve Buckland, who I'm going to talk about more a little bit later. But Steve founded CREEM around 20 years ago. So what we do at CREEM is we develop new statistical methods that are designed to help address pressing problems in conservation and in ecology. And in the time I've got today, I'm just going to talk about one aspect of that. So the problem I'm going to talk about is how many animals are there in a population. And the technique and tool I'm going to talk about is this thing called distance sampling. And I hope by the next half an hour, you'll come away with an understanding of what distance sampling is and how it can help answer that question: how many animals are there in a population? But before I do that, I'll start by giving some motivating examples about why it's important to know how many animals there are. So here's my first one. This guy on the left is called Ulas Karanth, and he was, until his retirement quite recently, the director of the Wildlife Conservation Society's India program. And Ulas has dedicated his whole professional life to conserving tigers in India. So you might think now I'm going to talk about how to estimate how many tigers there are. But one of the things that I'm not, one of the things that Ulas worked out is that in order to conserve tigers, the key things are you need to stop poaching of tigers, fine, but there also needs to be enough food for tigers to eat in their wild environment. And I'm showing two nice tiger food items here on the top right. So that's a sambar deer on the left and a guar on the right. And so estimating the amount of tiger prey is key to conserving tigers. And so in order to know whether your conservation work is going to be successful or not, you need to know how much prey there is in environments like the challenging environment below there, which is one of the protected areas in the Western Ghats. You can see this is not going to be easy to see where animals are and indeed to count them. Here's a second one a bit closer to home. These are, I'll pause for a second and let you guess to yourself. If you said harbour porpoise, then you'd be right. These are harbour porpoise. This is one of our iconic marine mammals around the coast of the UK. We need to know how many harbour porpoise there are and where they are, because there are lots of threats to the underwater environment, noise, pollution, and ironically at the moment, one of them is the noise from installing wind turbines, which is something that we have to do. But in order to know what kind of impact it has or is going to have, we need to know where harbour porpoise are. So this is a map of harbour porpoise numbers that came out from 2016 survey scans. This is the third scan, so scans 3, and scans 4 is taking place right now. So it gives us baseline for what's there, but also, you know, we can based on where wind farms are going to go, we can predict what kind of impact. This is one component of predicting what kind of impact they might have. Here's another example. So this on the right is a government publication it's one of the UK biodiversity indicators, and it's a composite of the numbers of farmland birds relative to 100 on the y-axis. There is sort of a baseline number in 1970, and farmland birds are as a group of around 19 species are one group that's declined quite a lot, particularly in the 70s and 80s as farming practices changed, some might say modernized a lot, but they've continued to go down since then. There's variation in performance among species. Some have been going up, but these species that have been going down quite a bit, sort of indicated by these sort of red bars at the bottom here, are particularly the ones that are specialized in farmland areas, including the 4 that I'm showing on the left there, which have been showing very strong declines. And so if we know which groups of species are declining and which are not, then we can target our management priorities towards those species that are declining the most. And if we want to see these in the future, we can obtain the best bang for the buck in terms of maintaining biodiversity. Okay, so this is the largest species that's ever been on Earth. It is a blue whale, right? You got it right. So I'm going to use this as just an example of counting things. So let's imagine that I wanted to count the number of blue whales. For example, they were over harvested from industrial whaling through to the 1980s. So maybe I want to know how they're recovering. And maybe I want to do that, let's say, in the Southern Ocean. So this is the ocean around Antarctica. Maybe I even want to use our smart new British research boat to do that. So what about if I just counted them all? So how would that go? Well, the Southern Ocean is around 20.3 square kilometers, and the boat may go maybe 150 kilometers a day. I can see around 14 kilometers to the horizon. So if I was just to go along in the boat and count every whale I saw out to the horizon, how long would it take me to cover the Southern Ocean? Well, around 13 years. So that's clearly not a feasible use for the Sir David Attenborough, or indeed even Boaty McBoatface. So we're going to need to shortcut that process somehow. And the way we do that is we take a sample. So here's an example here. These are little zigzag lines that you can barely see that were actually surveyed as part of a survey of Antarctica, not with this ship. And one key thing about if you're going to do sample lines, so if you're going to count things along lines, is they need to be representative of what's there in the bigger region. So this then brings us on to the question of survey design. How do you lay down lines and so on? And that brings us on to then what kind of plots, what kind of survey shapes are you going to use? So you could have lines, sure, like these ones here, which are systematically placed over the study area, or you could have little square quadrats, or you could have circles. So maybe some of you did some quadrat sampling when you were high school students, perhaps, and counted, you know, grass species or something in those plots. This is a diver counting underwater. This is a survey on the right here. This is of manatees in Florida from a plane. So all of these are examples of what we call plot sampling. So you put down plots using some design in an area, and then you count everything that's in those plots. Now, even this has a problem in some cases, which is illustrated in this picture perhaps, but better in the next picture. So here's a blue whale surfacing really close to my boat, and it's pretty obvious I can count it. But what about this one that surfaced a little bit further away from my boat? As you can see, it's rather difficult to spot. In fact, can you spot it? It's just there. So we're not necessarily, even if we do some plot sampling and put down lines through the Antarctic Ocean, let's say, that we're not necessarily going to be able to count all the animals that are in our plots. And so we need to deal with that and estimate the number that we're missing. So that brings me on to a method called distance sampling that I said I was going to talk about that deals exactly with this problem. And so in distance sampling, instead of having to have very narrow plots where you're sure you can count everything in them, you can have wider plots where we're going to estimate what we miss in those plots. And that makes things a lot more efficient, particularly for really rare species, because if I have to have really narrow plots, there's going to be quite a lot of things that I see that are just outside those plots. And that's annoying because I can't count them because they're outside my plot. So I'm going to throw away a lot of data. Whereas with distance sampling, you can have very wide strips or big circles, and then you can account for the fact that you're missing some animals. In order to do that accounting, we're going to have to gather some additional information. And the additional information that we're going to gather is the distance from where we are, from the line that we're on or the point that we're at, to the animals that we detect. So here I am going along a survey, and every time I detect an animal, I'm collecting a distance. And I'm plotting these distances in a histogram on the right here. And you can see that as this histogram builds up, we're detecting more animals close to the line than far from the line. And that makes a lot of sense, right? Because animals are easier to see when they're really close to you than when they're far away. So it's not that there really are more animals far from the line. It's that I'm detecting the easy to detect ones that are really close to me, and I'm missing the ones that are further away. So if I take that histogram, here we go, what I'm going to want to do with it is estimate the proportion that I detect. So, or you could say the probability of detection, because once I have that, I can correct for the ones I miss. So for example, if I detected, let's say 10 animals, and I know that the probability of detection is 0.5, that means that there were 20 there, right? Because I'm detecting only half of them. So this probability of detection thing is key. And I'm going to use my distances in order to estimate that. So the first thing I do is I put a smooth through this line because the little bumps and so on are just randomness. So there's got to be some method for putting a smooth through it. And then I say to myself, okay, if there are animals directly on the line, I'm certain to detect them. Because otherwise I would like walk right over them or something. I can't miss them. So let's say I'm certain to detect things right on the transect line. If I was detecting everything away from the line, because I put my lines down randomly or systematically or systematic random, it's not that there are less animals away from the line than close to the line. So if I was detecting everything, I should have detected the same number of animals at every distance. And it's this drop-off that's telling me about detectability. So it must be that these are the ones that I'm missing here. So if those are the ones I'm missing, these are the ones I detected, I can work out the proportions I missed as simply the area under this curve divided by the area of the rectangle, okay? Which I think in this case looks like about 0.7 to me. Looks like I'm detected about 70% of the animals, maybe 60, and I missed maybe 30 or 40. So you can see that if I can fit a smooth to this histogram, and if I can work out the area underneath it and work out the area of the rectangle, I can work out the probability of detection. And if you remember back to your maths classes, working out the area under a function is integration. So that's what we're doing mathematically, is we're fitting a smooth function and then we're integrating. Things are a little bit different in the point case. So this was the line case. In the point case, the geometry is a little bit different. So I go to a point and I get the distances to things going away from the point. And one thing to remember is that there aren't the same number of animals at every distance. So let me try and explain that. So let's look at the area of this circle here. So the area of that circle, well, let's say it's a radius 1, let's say 1 meter. Area of a circle is pi r squared, remember? So that's pi times 1 squared, so that's 1. So the area of this little circle here is pi. Sorry, pi 1 squared is pi, sorry. So the area of this circle here is pi. Now, what about this donut, the next one out, the next distance band out? What's the area of that? Well, it's going to be the area of this circle here, big circle, minus the area of this little circle in the middle, right? That's what's going to make the donut. And the radius here is 2. So it's going to be pi times 2 squared, which is 4 pi, minus the middle one, which is pi. So it's going to be 3 pi. Keep doing that for the next one. It's 5 pi, then 7 pi. So you can see that the area of these donuts goes up linearly as you go away from the circle. And if you're putting your points down at random, that means the animals are sort of randomly distributed in some sense with respect to where your points are, then the number of animals should go up linearly with each distance. So there should be, some number of animals here, and then 3 that number of animals here, and then 5 that number of animals in this ring here, and then 7 times that number of animals in this ring here, and so on and so on. So this is what your histogram would look like of distances if you detected everything. You can see there's a bit of randomness, but generally it goes up as you go away from a point. Now, if you actually do this in the real world, it doesn't look like that. It goes up for a bit, but then it starts going down again. Why is that? Well, it's because you're missing some animals. You're starting to miss animals as you get to bigger distances. And so this triangle is what it should look like if I detected everything. This curve is what it does look like when I start to miss things. So the proportion of animals that I miss is the area under the curve divided by the area of a triangle, so it's very similar to last time. Remember, it was the area of a curve and the area of a rectangle. This time it's a triangle. And I can work out the probability of detection at any distance if I want to know it. For example, at this distance here, so the triangle is about twice as high as the curve. So there are probably twice as many animals as I detected. So the detection probability is 0.5 at that distance. So I can work out the same kind of stuff as I was doing for a line transect. It's just the geometry is a bit different. Okay. This kind of ideas are really widely applicable. So here's a diversity of photos that I had on my hard drive from various surveys around the world. On the left-hand side are some marine surveys. So these are in polar waters here, surveying for whales. Here's in New Zealand. This one's in the Eastern Tropical Pacific. On the right-hand side, there's some terrestrial ones from, this is a helicopter survey of elephants. This is ice seals. This is polar bears. This is what a specialist survey plane looks like with very large windows. This is how you get distances out of the side of a plane. You can get them in bands. This is a survey for a terrestrial survey for tortoise in the desert in Arizona. This is for chameleons, plants, birds. This is kangaroos at night. So you can see a transect line that somebody's put out at night with some photos. And this is Ulas Karanth, the person I talked about before. And in this case, and in this bottom one, they're actually surveying the dung of animals. And then they can turn the dung estimate into an animal estimate. I can talk at the end about that if somebody wants to know more. And in this case, this is in St. Catherine's Protectorate. Mohammed here has got a person dragging a transect line in front of them, walking in a straight line, while they get the distances of dung that they see as a result from the line. He used to tie a rope to the back of a Land Rover and just get that going in a straight direction, but it hit rocks and went off course, and that didn't work very well. So now he's got a person that does it. Okay, so this is used extremely widely around the world. Now, you might ask, what has this got to do with St. Andrews? And the answer is Steve Buckland. So this is a picture, some pictures of Steve. This is a poster we made in his honor in 2020 at a conference that was about distance sampling and methods around distance sampling. Steve came in 1993, and he was already a pioneer in distance sampling in that time. He sort of pioneered the methods and then continued developing them since then. He founded CREEM, and a lot of what we do within CREEM is based on distance sampling. So Steve made many contributions, and they include sort of methodological contributions. So I'm listing some along the top. But for example, that smooth function that we use to fit, Steve did a lot of work on what kind of functions work well. He came up with that joint theory between line transects and point transects with the different geometry I was talking about earlier. What do you do with animals that run away from you? So animal movement. What do you do when you don't detect all animals on the transect line? Are there other variables you can bring in that will explain detectability in addition to distance? Can you produce maps of the density surface of the density across space, like that map I showed you of the porpoise earlier, and so on? So many methodological contributions. But the other thing that Steve did that we still do now is these methods are really not very much use unless people use them. It's all very well us as statisticians developing things, but it's vital that people out there use them. And so Steve really encouraged us and was sort of pivotal in producing user-friendly software, textbooks designed for practitioners, papers that are designed for different taxonomic groups that say, okay, how do you do this for birds? How do you do this for primates and so on? And a bunch of other related methodological contributions that sort of fill in gaps around distance sampling. And I like to think of this as a sort of a virtuous circle where there's some kind of a conservation need, and that makes us produce methods to address that need. And then once we've got the methods and we've published them and so on, so that we're sure they work, we do a whole bunch of knowledge exchange around that, which I'm going to talk about in a second, which results in the methods being used. And as they get used by more and more people and we contact those people, they bring up more conservation challenges, which inspires us to produce more methods and so on. And the other thing that helps with this loop and helps stimulate it is new technology. So it's very common, and I'm going to talk about that in a few minutes, that new technology enables us to survey things better. And we need to develop methods around it, but then we can use it. So let me talk about knowledge exchange for a little bit, and then I'm going to talk about new technology. So one vital component of knowledge exchange, and actually this is the reason that I came to St Andrews for a 3-year postdoc 20-something years ago, was to help with software development. So that's my main background, was in software. And so we produced a Windows-based version of the software, which is called Distance, that lets practitioners analyze distance sampling data. And it's developed a lot since then, over all the years since then. So it's now a sophisticated piece of Windows-based software. And I'm showing the analysis stuff on the left here. And it lets you do things like, for example, produce those maps and interface with the GIS fit lots of other variables to the detection functions and so on, the probability of detection, the things that influence that probability. But it also now incorporates a survey design component that brings in again a GIS and lets you lay out different shapes of transect line and see how well they're going to do. And I'd also like to pay tribute here. As I said, this is all a team exercise. So in particular, 2 people who are on the software development team. So Laura Marshall is one of them on the right here, who's now our main software developer, and Eric Rekstad at the bottom, who is the person mostly in charge of user support. And Laura has developed a simulation engine for the software now that lets people try out surveys in a simulation framework, which is much cheaper than doing it in practice, and see what's going to work, and then we can roll it out in practice. This Windows-based software nowadays has underneath it as an analytical engine scripts or packages that are written in software called R, which is this thing at the top, which is what more advanced users and statisticians use to do their analyses nowadays. And so those more advanced users don't have to use our Windows interface if they don't want to. They can go straight to R and do the analyses using this thing, which is rather less of a friendly package because it's command driven and you need to program it. But for advanced users, it's what they prefer. So that's the state of the software. It's been downloaded, Distance itself has been downloaded by well over 50,000 users now from well over 135 countries, which is a good portion of the countries in the world. And we have no idea how many people are using the R software because all of this is free and freely available to download, and the R packages are distributed in the crowd, and so we can't track how many people are downloading them. But there are an enormous number of publications and so on. For the Distance Windows software, as Tanya said, there's well over 300 people downloading it a month. And I'll show you some other user statistics in a second. So on to other aspects of knowledge exchange. So as I said, Steve produced these user-friendly books. There are 4 of them out now which describe the methods and sort of give advice on practice around that, good practice around that. We also hold a lot of workshops. So at the top here is workshops we hold here in St Andrews at the observatory where I'm standing now, where we invite users to come. Guess which of these people is from Texas, from all around the world, and come and learn both the basics of the methods, but we give also specialized workshops on different advanced aspects of it. And the funding, those workshops, unlike a lot of what we do, are not free, and we use the income generated by those workshops to help fund the software development. We also teach workshops in developing countries, and we get sort of separate funding for that. And so these are some photos I took on a workshop I taught in Tanzania a few years ago. And they're very different in format. They involve quite a lot more of a practical aspect to them. So this was at a field camp, and this is sort of the first day practice in a very artificial framework, and then classroom time and computer time and so on. Since COVID struck, we've moved a lot of our workshop teaching online, and so we teach sort of small group live workshops online. We've done over 30 of them since 2020 now. But we also have, those are paid, but we also have recorded all of our lectures on the introductory workshop and made it available for free in an online format. Also, for all of our paid workshops, we have a widening participation scholarship scheme. So we give away at least one place per workshop and have sort of a scholarship system for that. And then the last aspect of knowledge exchange is internet support, both for the methods and the software. And this is the bit that Eric Rekstad is in charge of. So we have a website where you can download the software and so on, but we have a very lively email list with over 1,000 people on it. And I just printed this out yesterday, and there's been, already quite a few queries, but also responses over the last month. Okay, so I want to turn to new technology now. So I'll cover a few and then talk very speculatively on my last slide about the future. So the first one is camera trap methods. So now these wildlife cameras are quite ubiquitous. They're sort of infrared driven. As something goes past, they take a photo. And those are used for nature reasons and so on, but we wanted to develop methods to use them to actually estimate the number of animals in an area. And this was work done by Steve Buckland jointly with a PhD student, Eric Howe, who's at the Ontario Ministry of Natural Resources. And so by when you lay them out, by spending some time measuring distance from the camera and having some distance signs like this that you just show on either the photos or some of these take videos, then as animals come past, you can determine what distance those animals are at. And so it's a bit like one of those point-based distance surveys that I talked about earlier. It required some adaptation of the methods. We've then, under funding from Wildlife Conservation Society, led by Sam Strindberg, and jointly with Noemi Capel and Eric Rekstad, we developed some workshops around the software extensions, and now we teach workshops on camera trap distance sampling. And this method, particularly for forest animals, where they're very hard to see, particularly in tropical areas, is becoming the standard method now. Another aspect of new technology is acoustics. So there are lots of animals that are very hard to see, but they're easy to hear. So this is a beaked whale. It spends most of its life kilometers under the water, comes up very rarely to breathe, but it makes echolocation clicks to find its food because it's completely dark where it lives most of its life. This is a blue whale, as I had before. Blue whale songs can be heard for hundreds of kilometers underwater. A gibbon that sings, and a forest elephant. Forest elephants make infrasound. You wouldn't think an elephant's hard to see in a forest, but they are, but they make long distance sounds. So we've been working for a few years on methods that enable you to go from the sounds you might record on a remote microphone or hydrophone coupled with a recorder into an estimate of how many animals there are. This was started with a couple of colleagues in the States who were both acousticians, and has been done, from my perspective anyway, jointly with a couple of colleagues here. There's other people at CREEM also who are working on other methods. Tiago is sporting a very nice tag on his head from a recent meeting, as you can see, suits him. And Danielle, sorry, my first colleague here, Danielle Harris, got into this because she was working on her PhD, and it was inspired by this guy, Luis Mateus, at the University of Lisbon, who is a seismologist actually, and had a data set. This is Portugal here. This is Gibraltar. A data set of underwater seismometers in the near Atlantic here. And he was a bit puzzled because he could hear this weird call or sound on his seismometers, which this is very low frequency. So there's frequency on the y-axis here and time on the x-axis. And he stood up at the conference and said, look, I have this sound, I don't know what it is. And somebody stuck up their hand and said, I think it's a fin whale. As a person working on earthquakes, which was his interest, and tsunamis, he had no idea. But we ended up working with him and working out a method for getting the distance to those calls. And when you do that, they look like this, which is exactly like a point transect. So we could get call density, and then with an estimate of how often they call, you can get animal density from it. We've been working on these methods a while, and my favorite survey here is one that we did on harbour porpoise again, but this time in the Baltic Sea, where there are very, very few harbour porpoises, and standard visual line transit methods from boats or planes just do not work. So the last plane survey of harbour porpoise in the Baltic detected one, and you can't do really anything with a sample size of one. So we developed an acoustic survey method funded by the EU, where we put out 300 acoustic sensors across the Baltic for 2 years and used that to get an estimate of this very small population of Baltic harbour porpoise. This is joint work with my colleague Louise Burt here, and 3 superheroes from various parts of Sweden. It was a joint project with all the surrounding countries in the Baltic, apart from Russia. And what we found at the end of this was a breeding population of harbour porpoise that are in this area. We think there's around 500 porpoise there in the whole Baltic. And as a result of that, that area was set aside by Sweden as the largest Natura 2000 protected area that had been set aside at the time. So it's a real conservation success. Okay, so in the last couple of minutes, I just want to talk very speculatively. I just have 3 bullet points just to raise some sort of new technology that I think is coming down the pipe that I'm really interested in working on some more. So the first is with camera traps, the way they work historically is that somebody has to go through all those images, work out what animals are there, and then based on comparing it with those previous images that had somebody standing there with numbers showing how far away things are, they enter the distance for each of the animals they detect. And clearly this is a job now for automated image recognition, which is becoming more and more of a thing. Those of you who know anything about machine learning and image recognition will know this is becoming very powerful now. And the other thing that's starting to become quite powerful is ranging. So by just looking at the patterns of and sizes of things that are on the image, you can guess distances to them. So that's an area in collaboration with computer scientists that is a sort of a hot area. And I think that's going to really increase the potential for using these camera trap surveys in tropical areas. The second that's a little bit similar, but now is in the acoustic realm. So those acoustic detections or acoustic systems that I was talking about before, they already mostly use automated detection and classification mechanisms. So you have enormous terabytes of sound data that you've recorded. A machine goes through it and picks out the things that it hears and classifies them and so on. Then there's some ground truthing that goes on. And the ability to do that is getting better and better. But the thing that isn't automatic right now is ranging. This is, I've done most of my work in the marine environment, but the thing I'd be really interested in is in the terrestrial environment. At the moment, a lot of songbird surveys are still done by humans who go into the forest and they stand at a point and they detect birds, and then they work out what distances those birds are by just guessing effectively. I mean, using their expert knowledge to know. I think there will be a lot of room to develop a sort of little fold-out 4-element microphone system that you could just stand there with it or leave it there for a while, and using time difference of arrival and other cues, it would work out what the distances are to animals, and it could do either at the time or post-processing some automated detection. I should have said earlier for the previous one as well, you could do automated ranging based on the picture itself, but the other obvious thing to do is to use exactly the same technology that's built into autofocus in cameras to actually get the distances to things using LiDAR or acoustic methods, or exactly the same kind of variety of things that cameras have built into them. And then the last is really speculative, but I wanted to mention it, particularly because Tanya was talking about it, is this idea of using environmental DNA to estimate animal numbers. So this is kind of your crime scene investigation type level stuff. So we all leave DNA, as you probably know from watching cop shows, around wherever we go, and animals are just the same. So DNA sloughs off, let's say, these dolphins in the ocean as they go along. They also poop and they breathe, and all those other things leave DNA signals behind them. So can you use the amount of DNA that you get for animals eventually in order to get an absolute abundance estimate for those animals? And you can imagine that the further away you are in space from where those animals were, because DNA spreads through the water, and in time, because it degrades over time, the less likely you are to detect, or the less of it you will detect. And so it sounds a little bit like distance sampling, right? Where the further away an animal is, the less likely we are to detect it. So we're starting on a project jointly with the University of Washington, and this is we. When I say we, it's my colleague Erin Jacobson and I, to see jointly with them whether there's any mileage in using these kinds of distance sampling ideas in order to be able to estimate absolute animal abundance. I think we're quite a few years away from being successful with that, but it's a brilliant idea. And if it works, it would mean that people could just, for example, in the sea, collect seawater samples regularly, and we could use that while they're going on ferries or anything else, and we could use that to estimate animal numbers and then have these conservation benefits. So I think it's got a lot of potential. Okay, I wanted to leave you with this picture, which is, if you come and visit me here in St Andrews and you come into the observatory, you come in through our front door and look left, there's a mural on the wall. It's about, it's taller than me, 8 feet high, it's probably 20 feet long, and this is what it looks like. And on the mural, apart from a watchful researcher in the middle there, are pictures of some of the animals that we work on. And if you look hard, you'll see some of the formulae as well. And these are formulae we use in doing our work. And this is on a website, creemural.org. So you can go to that website, and if you do, you can click on the animals or the formulae and find out more about the work we do. So if you're interested in what I've been talking about and you want to find out more, please go to that website and find out. And you feel free to contact me anytime about anything I've said. Okay, I'll stop there for now. Thank you for listening.
Thank you so much, Len. Okay, we'll jump straight into questions. So a big one. Your talk is about estimating population size, but do the numbers really matter? I mean, isn't it usually obvious what the right conservation decision is, and it's other factors like politics that stop us from getting the right thing done?
Well, okay, so that's a hard question to start with. So a few things to say about that. So one is that I can't think of a case where a worse decision was made by having better information. So it's true that sometimes politicians or other factors, override what's right for animal conservation. But making an informed choice has to be better than making an uninformed choice. And secondly, no, it's not always true that that's the case. So it's quite often the case that the right decision is not obvious at all. So, for example, if we want to invest our money in Britain on biodiversity conservation, it's not obvious that farmland needs more investment than shorelines, rivers, etc., urban environments, for example. So it's really quite common that the numbers that we produce really do help people to make better real-world conservation decisions.
Thanks. Okay, lots of thanks for an interesting talk. Is distance sampling assuming that the density of animals across the sample area is uniform? And if so, how do you know when that is a valid assumption for the area being measured?
Oh, that's a brilliant question. Somebody's really very alert, so thank you for that. No, we don't assume that. So there is an assumption in the methods, and that's why it's a brilliant question, that the number of animals, the true number of animals, let's say one of those line transects, the true number of animals is the same at every distance. And so you might say, okay, hang on a second, you're assuming that the number of animals is uniform, but I don't actually assume that. What I assume is that the transects are random. So it doesn't matter what the distribution of animals is. If I put transects down at random, I'm randomizing my position with respect to the animals. And so there is going to be the same number of animals on average at every distance from me. So that wouldn't work, for example, if I did line transects down the middle of the road, then there's no reason to suppose there are as many animals in the middle of the road as there are in the forest just to the right or left of the road. So you need to have random lines in order for this to work. And in particular, so when I've got random lines, the other thing I can do is, if I'm interested in the spatial variation in density over my study area, is I can model that. And that's what ends up producing those density maps that you saw earlier. So that's a really interesting inference that we can get out, is where are the high density areas and the low density areas.
Yeah, and you mentioned the assumption that animals at zero distance are detected, which seems reasonable for those deer in India. But what about the whale, where a ship can go over them without detecting? People
are really paying attention here. Okay, so I did skip over that, and it's a really important assumption. So it's the sort of fundamental assumption of what we call conventional distance sampling, which is the early stuff. And as you said, it's perfectly valid in many situations if I'm surveying many terrestrial animals and some marine animals, because even though marine animals dive under the water, if I'm going in a ship, then that animal is likely to be up at some time while I'm going past them. So if it's right in front of me on my transect, then I'm likely to see it. But for example, those beaked whales that I talked about, they can dive for well over an hour. And so it's not infeasible that while I'm going past, then the animal's not available to be detected. So what you need to know in that case is what's the chance that the animal's going to be at the surface at some point while I'm going past. So you need additional information about the dive pattern of that animal. So that's one answer, is that we need separate information if it's an availability problem, so the animal's not available to be surveyed. But the other thing that can happen is that it's not actually true that all animals that are available are always detected at zero distance. So an example might be those elephants, those forest elephants. If I'm flying, or savanna elephants, let's take an easier example. If I'm flying over them in an aeroplane, if they're sort of hidden underneath a tree, I'm not going to detect them. They're in theory, they're available because I could, if I looked hard enough, see them, but I just missed them. And so it's not that everybody's going to miss them. It's just that the chance of seeing them is not 1. And in that case, what you can do is extend the methods to include a second observer who's independent of the first. And there's information then in, so effectively what's happening, imagine I'm the first observer and I detect that animal. I'm setting up a trial for the second observer who's behind me. Do they detect it, yes or no? And the proportion of those animals they detect tells me about detectability. That's actually a separate survey method called mark recapture. So effectively, I'm marking it with my eyes, and then that person may recapture it or they may not. Mark recapture is also a well-known way of detecting animal numbers, of surveying animal numbers. And in fact, David Borchers, who was the director of CREEM, gave a talk on that in the Saints talk 2 years ago, which I highly recommend going to. It was a very nice talk explaining those methods. And David himself, for his PhD a few years ago now, combined mark recapture with distance sampling by this double observer, independent observer method. And so, for example, in shipboard surveys on very big research vessels, that idea of having multiple independent observers, maybe one high up in the crow's nest and another on the bridge, is a standard way of getting around at least part of this issue. So not the availability part, but this issue of missing things right on the line. And yeah, it's a very well known and well used method. So thanks for the question.
So three more questions on methods. First, okay. Do you do any testing to understand how accurate your estimates are?
Yeah, great question. So there's two types of testing you can do. So one of them you're not going to be very impressed by, but it's important, is just a simulation study to say, if I generate perfect data, do I get the right answer from my fancy statistical method and from my software? And then if I put problems into the data of various kinds that the methods wasn't designed for, how robust is it to misspecification? And that's a really important part of checking how the methods are going to perform in the real world. But it's not a substitute for the other thing, which is how do they perform in the real world? So there have been quite a few studies where we know the true population size because we're working in an enclosure or some other reason why we know, and we try the distance sampling methods on those. And in the situations where that's done well, it works really well. The times that I've seen papers published saying distance sampling doesn't work are, well, all the ones I've seen are cases where people basically aren't using the methods well. And there's been a few times, particularly Steve's led on some of this, and my colleague Tiago, where we've ended up writing papers saying, well, the problem was you weren't using the methods very well, here's how to do it better. But I don't really like doing that kind of thing because it's very confrontational. I think a better thing to do is to focus on how to use the methods well and training and explaining how things can go wrong. And in general, what we see is that the practice is getting, the level of that people are working at and practice is getting better and better. So I feel pretty confident that we can diagnose when things go wrong from the data or from knowledge of how the survey was and predict when things are going to produce reliable results or not.
I want to just divert quickly because you talked a lot about the strong case for knowledge exchange and all of that training, and just wondered, where do you get the funding for that?
Yeah, that's a difficult question. So we think it's really important to do this outreach, to have our methods used. In fact, that's why we're in this area in the first place, but it's really difficult to fund. So we get research funding to do the research, and often we work with practitioners to do that. But then supporting, for example, the software development, we've done it historically from workshop income, as I said, but we're finding with COVID and other things that fewer and fewer people are willing to come to in-person workshops. We also appreciate that they're really expensive for somebody to travel halfway around the world to come and live in St Andrews for a week to come to one of our workshops. And the online workshops, the expectation is much less that you're going to pay a lot of money for that kind of thing. And so, and plus, you know, we took the decision to give our introductory workshops away for free just to increase participation. So it's a real struggle, to be frank, to fundraise to provide ongoing development and support for the software.
Right, a call to action.
Yeah.
Okay, so there's a question here. Is there a method to consider hard boundaries, i.e. shorelines for marine animals? Yeah,
that's a good question. So yes, there is. So it really depends on whether there are any animals on the other side of that hard boundary. So if, let's say it's a fence and you have no reason to suppose the number of animals is different from one side to the other, then your survey line will have to stop at the fence and can come out the other side. But you allow yourself to detect animals on the other side, and you'll get an average density close to the edge that's about right. But if there aren't any animals on the other side, then you're going to you need to make sure that your survey coverage close to the edge is even, or is the same as the survey coverage near the middle. And so there are special sort of survey design adjustments that you can make to make sure that if you have to break off a survey line when you get to the edge, that you add a little bit of survey line close to the edge somewhere else to make up for it. And the software is programmed to allow you to do that kind of thing. It's a little bit annoying to do in practice, I have to say, because you end up doing small bits of survey line, but it makes sure that you have sort of unbiased estimates overall.
Okay, quickly, do you assume the movement of the ship or observer is independent of the movement of the animals? Like with some species, this might not be the case, like dolphins following ships.
Yeah, that's a really good question as well. So, so the basic method is a snapshot. It doesn't allow animals to move at all. So basically, the animals and humans are fixed in space at each sort of instance as you're going past, and you get the distance to them. Random animal movement causes relatively little problem as long as the movement isn't very fast, but relative to your movement. But in the case where animals run away from you or are attracted to you, clearly there's a problem. And one approach to that is to try and detect them before they respond. And so if you have this 2-observer protocol with one person up in the crow's nest, they're looking very far ahead and trying to pick up animals and set up trials for the other person before those animals respond. That's one possible approach. But if you really have a pathological problem with animals responding a lot, then that really isn't the right kind of survey method. You need to think about an alternative approach.
Thank you. Okay, I don't really understand this question, so I'm going to read it out. How do you know how accurate the estimated distance of the target animal is?
Yeah, okay, so they're asking about like ranging. Can we tell how far away the animals are? But we've done quite a bit of work on measurement error. So does it matter if you get it wrong? And in the line transit case, it doesn't matter a lot as long as your error goes up as the distance goes up. In the point case, it does matter. And so it becomes very important to get the measurements right, or to characterize what the measurement error is. And in that case, you can build it into an extension of the methods. And in the terrestrial case, the standard way to get distances is with a laser rangefinder, if the animals are sort of at that kind of appropriate distances, like not too close. If it's really close, you can say that's where the animal was, and as you go past, you just measure it with a tape. And measurement methods are, you know, a sort of very valid way of doing it, at least if you can say, yes, the animal was there. You don't measure to the animal because it's long gone, but if you can say where it was. So I think measurement error is not one of the worst problems that we typically have. In the acoustic case, when we're trying to get measurements sort of by guessing from time of arrival at hydrophones or microphones, it can be a bad problem. And we need to build in these measurement error methods as a sort of add-on to our standard methods. Yeah, good question.
So a question around the relatively new field of airborne eDNA. How to estimate terrestrial species population numbers from the data collected using these techniques and the challenges faced. Have you conducted any work?
No. So we're just starting on eDNA work, and I am not sure at this stage it's actually going to work. It seems like a really heavy lift to me. But in the in the marine environment, which is the one I'm working on in the project that we're working on at the moment, There are several prongs to the study, but one of them is working in the Hood Canal in Washington State with a population of bottlenose dolphins that are actually there for the US Navy. And bottlenose dolphins are not native to that area at all. So the only bottlenose dolphin DNA in the Hood Canal comes from those dolphins. And we know where those dolphins are. So we can take samples at various distances away from those dolphins, and we know where they've been as well within the area that they go, and see how their detection numbers fall off as we go away. So we can sort of calibrate a detection function with them. So also what we're doing alongside that is we're using particle tracking in the water to try and predict how the DNA from those dolphins, we know how much DNA they're shedding, or we're measuring how much they're shedding. We're trying to predict where it goes, and we're seeing whether those sophisticated models can match what we measure. And if that is the case, then there's some mileage perhaps in doing the same kind of other studies of like modeling it, rather than, because you can't measure it normally because you don't know where your animals are, but modeling how it might disperse in other environments. So sort of baby steps, basically. In the terrestrial environment with wind and so on, I imagine the process is very similar. You know, we have ocean currents here, there are air currents, but it's not something I know anything about. And if the person asking is sort of interested, then please contact me, and I'd be happy to chat about it some more.
Great. Watch this space.
Yeah.
In distance sampling, do you estimate the detection probability for each transect, i.e. refit the histogram for each transect?
Yeah, that's another great question. Fantastic questioners. So if you're just interested in the average density over the study area, then there's an absolutely magical property of distance sampling called pooling robustness. And what pooling robustness does is, we know in the real world that detectability varies by lots of things, like each animal probably is more or less detectable, each habitat within your study area has different detectability and so on. But if you pool all those detections together and you just fit one of these functions, turns out you get the right function on average, as long as you're detecting everything at zero distance, which is the standard sort of assumption in terrestrial cases. So we don't care really, as long as it's not huge variation, where the detectability varies. And so we just fit one pooled function. Now, what about the case where you're actually interested in variation in density? You want to produce one of those density maps through your study area. Then obviously, if you don't account for the fact that animals are more detectable in one area than another, then you're going to think there are more animals there just because you detected more of them. So you need to account for this. And that's where we bring in additional explanatory variables into our modeling. So we don't model each transect separately, but what we do is allow additional variables like the habitat on each transect segment, the observer, the weather conditions, the group size of animals, all those things to come into our modeling. And it becomes a more sophisticated modeling approach, but what you get out of it is you can predict what the detectability is in every part where you surveyed, and so correct for this variation that way.
Thanks. So a question about being online, which we are all doing a lot of online learning, for example, this talk. For the distance sampling training that you do and the international audience, what do you think and what do you find the pros and cons of doing it online versus in person are?
Well, for one thing, there are obvious cost differences. So, you know, and I've always felt this is an issue with our online workshops, which are brilliant. It's wonderful to be able to meet, you know, people who use the methods and work with them for a week, you know, sitting side by side with them on their studies and so on. But they've had to travel halfway around the world to be here with us, and spend a lot of money to stay in St Andrews and burn a lot of carbon to be here. That's a huge advantage of being online. On the other hand, it's really hard for them to spend a week. You know, when they come with us and on our workshops, we spend a week together in a room basically, and we eat, sleep and talk distance sampling, and they go away with sort of all the knowledge they've gained and also all the other people that they've met on that workshop. Whereas the online experience is very different. You can't do it for a full day. I mean, our segments are much shorter, they're just a few hours. And although people interact with each other somewhat, I think that the whole group dynamic is very different. So in general, the experience honestly isn't as good, I don't think. But the participation, I mean, we've had, Eric's produced a very nice map of the people that have been on his live introductory workshops, and they're from every continent. The last camera trap distance sampling workshop I taught had people from every time zone. You know, we had real trouble finding a time of day. It was 3 in the morning in Australia. There were people from Southeast Asia, from West Coast of America, East Coast, from Europe, all at the same workshop. And so there's, I guess, an advantage of sort of democratization in some way, but a disadvantage that the experience isn't as good.
Thanks. And final question, if you were advising a young person keen on getting into conservation, what would you tell them?
Well, so I'm biased, so, you know, take my answer with a pinch of salt, but my experience is that there's lots of people who want to get into conservation, and it's brilliant that people do, but it comes as a real shock to a lot of people that it's a quantitative science. It isn't, I mean, there's a lot of it about social science and so on is really, really important, but conservation itself is a quantitative science. And so I would say study maths at school and make sure you get a good quantitative training as part of your learning about conservation. There is another slight advantage to that too, which is that if you're really into conservation, it's actually quite hard to get a job in that field because lots of other people want to get a job in that field. There really aren't that many people like us here at CREEM. We're sort of lonely in a way. There aren't that many people who are into mathematics and into conservation, because most people, at least who are into statistics, they go and work in the pharmaceutical industry, or internet gambling is a big thing at the moment, and earn, you know, lots and lots of money doing that. And as I said, the people who are interested in conservation, most of them, a lot of them, are not really interested in quantitative science. And so it's actually, you know, come and get a job with us. It's a lot easier to get a job, I think, if you're interested in conservation with us. And then lastly, you know, I hope I've shown that you can be really effective and make a real difference by applying quantitative science in this field.
Professor Len Thomas, thank you so much. It's been fascinating. Thank you everybody for coming. Join us for our next talk on Thursday, 22nd of June during Learning Disabilities Week. Dr. Sylvia Paracini's talk is From Genes to Brains, Multidisciplinary Approaches to Understanding Dyslexia and Specific Learning Difficulties. Thank you very much, everyone. Good night.